Good afternoon. I'd like to share with you the incredible journey that we've had here at Dyadic over the last 41 years, from the blue gene business to the genetic genes, turning DNA into life-saving medicines more affordably, in larger volumes, more rapidly, and how we're using this technology that we spent 25 years to make enzymes at massive scale affordably to help with the pandemic, to be better prepared for future pandemics, and our main focus has been on developing and mass producing and helping the pharmaceutical animal health companies develop and produce more affordable biologic vaccines, drugs, and other therapeutic proteins. Here at Dyadic, we are using a platform technology that we spent over 25 years developing. There's been several $100 million spent between DuPont, BASF, Shell Oil, Abengoa Bioenergy, and Dyadic. And we're leveraging what we learned in the industrial enzyme business, where we sold that technology to DuPont for $75 million on December 31st, 2015. And since then, we've been focused, laser focused, on modifying this cell line to be able to produce quicker, faster, lower cost antibodies, vaccines, and therapeutic proteins for animal and human health. We're working with all four of the top four animal health companies today, an additional global animal health company. We're in discussions with others. We're working with top 5, top 10, top 25 global pharmaceutical companies. There's a lot of people test driving the car, so to speak, or the cell line, working on it to find out what it's capable of doing, what it's not capable of doing, what we can make it do that it can't do today. We've spent a lot of money on our own internal programs on modifying that cell, removing the protease genes that encode for protease activity that were degrading some of the cells or the proteins that were performed or produced in those cells. You'll see coming up that we were very successful at doing that and eliminating that background that led to hyperproductivity. We're working on glycoengineering the cells so that we can make monoclonal antibodies, because our antibodies, just like CHO cells, they bind and neutralize virtually identical. People often ask, what's C1? Well, it's an easy answer. C1 is a hyperproductive filamentous fungal cell that came about through serendipity, through a serendipitous random mutation that led to hyperproductivity and low viscosity. That led to mass production at low cost, at flexible commercial scales, and that led to the ability to then develop a recombinant protein host, which means we can take DNA, put it in, take DNA, put it out. If we want to make something at higher levels, we put in gene copies, we put them in a site-specific integration site that we learned after 25 years and several $100 million of research in the industrial side. We can make stable cell lines very quickly. They're high productivity. We can then optimize the fermentation conditions and push that yield even higher. We grow it up under very low viscosity that came from that serendipitous mutation. So how do you compare this to what's existing today? It's like night and day in some cases, and we'll get into that on the next few slides. When you look at how we can make an antibody, we've made one at 24 grams per liter in 7 days, or 3.5 grams per liter per day on glucose. Faster, quicker, cheaper. We've made 300 times more of an antigen versus baculovirus in a ZAPI program in Europe, and we'll talk about that upcoming as well. Before the pandemic, we wanted to disrupt biomanufacturing. Now, as a global society, we have no choice. We need to change the cell lines in the tanks. If we keep doing the same thing over and over again, expect different results, we're going to get just that. No different results, too high, too little, too late, just like we saw in vaccines. And monoclonal antibodies. We hear about Regeneron and the miracles through President Trump, but the doses and the quantities that are needed, the amounts, the cost, it's unsustainable, it's unaffordable. With C1, you'll see we can make 4 times the amount quicker, faster, cheaper, and we'll talk about that. High yield, secreted proteins, low media costs, shorter development time, the unique morphology that led to hyperproductivity and low viscosity, and we can change the fermentation conditions very broadly, very widely, both temperature and pH, to achieve the goals and objectives that we need to make large volumes of low-cost proteins for animal, human health, and to defeat the pandemic. In biotech, it's all about the data, and we have the data. The data is amazing. People have to wonder why this cell line hasn't been adopted and used more widely. And the answer to that is people haven't known about it. It was an industrial biotech cell line that is relatively new to the pharmaceutical and human and animal health. If you think about it, and you look at the facts, and the data spells it out very clearly. In the Zoonosis Anticipation Preparative Initiative, which is called ZAPI, which is an EU zoonotic disease to humans that we've been involved in since 2015. We've demonstrated in that ZAPI consortium that we made 300 times more of the SBV antigen that came from the second place cell, which is baculovirus. That's an amazing difference. The Rift Valley fever virus antigen, we made it at extremely high levels, couldn't be made at all in baculovirus. And then when you look at Sanofi Pasteur and the data from the seasonal flu, and you can click on the link in our presentation or find it on our media center under presentations and scroll all the way down, we not only made more of it, we used 1/3 the amount and got a better result in the mice trials. And those aren't our data, that was Sanofi's data, which you'll see on those slides. This is from Sanofi itself. It was safe, it was effective, and at least as immunogenic. And the at least as immunogenic is an understatement. And I think you'll see that when you look at that presentation. One of the most important data points is one of the recent data points. When the pandemic hit, and we had been involved for 5 years with the ZAPI group in Europe on zoonosis diseases, with the SBV, with Rift Valley fever antigen. We worked on MERS as well in that program. We were working with the Israeli Institute of Biological Research on an enzyme to combat VX and sarin gas, to again develop a platform to be ready to be affront pandemics, epidemics, and other biological threats. We took the gene sequence from the SARS-CoV-2 receptor binding domain protein and programmed C1 with it, and we're hitting record levels that people can't believe. And not only cannot believe that we can do the high levels of productivity at 3 grams per liter, it's in 5 days, and we grow it up on glucose and very low-cost media. We can use this cell line and R in a variety of projects all over the globe to help combat this horrific disease by helping to make billions of doses for pennies on a dollar in some cases compared to other technologies. As I mentioned, we produced this receptor binding domain protein from the SARS-CoV-2 spike protein. Today, that protein is being evaluated and is running in 7 different programs with 10 different animal studies that we expect will be completed by year-end 2020. In addition to that, we're working with the Frederick National Laboratory on their gene sequences, programming C1 with genes encoding vaccines of interest to them, Ufavax, which is a Scripps spinoff, and we're discussing with a variety of other parties as well. In addition to working on vaccines, we're also working on monoclonal antibodies, and there's been even more attention since President Trump has been using the Regeneron product to potentially use C1 to make monoclonal antibodies, and we'll talk about that coming up. With C1, we can not only make large amounts of low-cost vaccines that are safe and effective, but we can make large volumes of monoclonal antibodies and other therapeutic proteins. So what does that mean in a COVID world? It means saving lives. It means lowering cost. It means access. It means affordability. It means scale. We can run 4 batches of a C1 monoclonal antibody product in the same time it takes to run 1 batch of CHO. We can get higher yields. So if you can get higher yields, you make 4 times as much in terms of quantities because you're running 4 batches. Each batch produces more. The cost of the media is a fraction of what the cost is for CHO cells. We have no choice. The world cannot make enough monoclonal antibody treatments for the amount of people in America, let alone globally. We have to do something. We have to do it differently. We have to do it better. We can't keep doing the same thing over and over again and expect different results. We're just not going to get it. I mean, that's Einstein's definition of insanity. We need to change the cell line in the tank if we're going to make a difference. We need faster, more efficient, hyperproductive cell lines. And we've spent the last 25 years developing just such a cell line. And for the last 5 years, we've been tweaking it and modifying it to fit to make animal and human health products. And we're doing that, but we're also here to help combat the pandemic. Again, it's all about data. This data is amazing. These are record levels on virtually every front. When you can make 3 grams per liter per day, when the average of a monoclonal antibody is 4 grams in 14 days, that's a huge difference. When you can do that on glucose and inexpensive chemically defined media versus very expensive CHO media. So you can make more batches, you can do it quicker, you can do it for less money, you can save more lives, you can treat more people. We need to change the cell lines in the tanks. We don't need to replace CHO, we need to supplement CHO. And if we don't do that, people are going to die, they're going to suffer, and we as human beings can't let that happen. And we're asking the industry, the governmental agencies, and academics to help us speed the adoption and use of a cell line that's not new. We spent 25 years developing it to make massive amounts of low-cost enzymes in the industrial world. We're just refining it to make it fit in the needs of animal and human health, and now that the pandemic hit, to help eradicate the COVID-19 virus. Again, what are we trying to accomplish? We're helping respond to the pandemic of today, be better prepared as we have been with ZAPI for the last 5 years, with the Israeli Institute of Biological Research since January 2018, working now with the Frederick National Lab and others to be better prepared for the next pandemic, in addition to the one we're faced with right now, because surely another one will come. In addition, the goal here is there's a silver lining. The technology, taking this technology through the FDA, getting an approval for a vaccine, an antibody, or a therapeutic drug, allows us to then help drive down the cost of prescription drugs for the future, so that we can make healthcare more accessible and more affordable to patients globally. We have very experienced board members that come from big pharma. We have executives that come from big pharma, small biotech, and they're all driven. And we're driven for one purpose, and that is to make healthcare accessible and affordable to patients globally. And in order to do that, we believe we need to change the cell lines that are being used to more efficient cell lines that grow faster, produce more, and cost less to run, that can be used at flexible commercial scales. So some of the messages that I'd like you to leave with are the following. First, we're well financed. We had $32 million after Q2, and we only burned through $4 million in the first 6 months of the year. So we're well financed for many years going forward. In addition to that, there's no warrants, there's no debt, so we're in great shape. We've got probably the cleanest cap structure of any of the small biotech companies on the NASDAQ. In addition to that, we have driven, successful, proven management teams that have created value for shareholders. In fact, between me and my children's trust, we own roughly 30% of this company. So our interests are aligned with shareholders. They're not in the opposite ends of shareholders. We protect our shareholders. Why? Because it's the right thing to do, and of course, we're protecting ourselves at the same time. But we're making great decisions, guided by experienced executives, from our executive team to the board members to the world-class scientists that we work with. I would suggest you go to our website, which is www.dyadic.com. There's some videos that have been recently released on the technology, some animation that shows you what we do, how we do it in an animated form. There's a collaboration we have with the Finnish government lab called VTT Technical Center, where there's several scientists that we work with there that are world experts at engineering fungal cells. So we go to the places that we need to go to create the value. And we do so efficiently and effectively, watching every dollar like it's our dollar, because 1/3 of that dollar really is our dollar. Thank you for attending, and we look forward to working together with you.